Right. This presentation covers the urinary system. So let's get started. All right, we talked about this case in class, but just to briefly cover for this presentation, let's say that we have a 3-year-old male-neuter domestic short-haired cat that presents for stranguria, hematuria for the past 2 days, and now it's led to anuria. On presentation, the patient presents painful, vocalizing, tachycardic, and has a mentation of just being very lethargic. On palpation of the abdomen, you realize that the abdomen is very hard and possibly the urinary bladder is very large. Looking on an ultrasound, you're able to confirm, yep, that abdomen is pretty much just a urinary bladder and it's huge, full of urine. We also could see a snow globe effect. That might mean that there's mineralization and crystals and buildup in the actual bladder, which is not normal. We don't want that. So what could be going on? Well, we talked about how this could potentially be a male blocked cat. And the blocking is actually due to crystals, and they're going to form and create sediment, and that's going to also form stones and ultimately some uroliths. So looking at this picture, we can see that these are struvite crystals. We'll get more of this in Lab Techniques 1. And then we can see calcium oxalate crystals. Remember I talked about with the calcium oxalate that yes, we can see these for various reasons, but we can also see it if our urine sample has been in the refrigerator. So it's important to know when and where your sample came from and what time of day it was collected. Now, over time, these crystals can produce uroliths that are going to be the urinary bladder stones. Here's an example of an X-ray and then a bunch of stones that are pretty much encompassing the entire urinary bladder. Okay, in this presentation, our objective is to talk about the functions of the kidney. Also, what does the kidney look like? And how does blood come into play and flow into the kidney? And then where does it go? And then how is urine produced and by how much? And how the kidneys help with our blood pressure and how the ureters, urethra, and bladder are all connected to our kidneys in some sort of way. So kidneys, what do they do? Well, one, they're going to remove waste products from the body. They're going to remove drugs from the body. They're going to balance the body's fluids. They're going to help release hormones that regulate blood pressure. They're also going to produce an active form of vitamin D that promotes strong and healthy bones. They're also going to control the production of red blood cells. Now, how they do that is by a hormone called erythropoietin. That's very important that you know this. It's a hormone produced mainly in the kidneys, and it's going to stimulate the production and maintenance of the red blood cells. So, it's important for us to talk about waste management when it comes to the kidneys. The urinary system is the single most important route of waste product removal in the body. So from the kidneys and the urinary system, we get homeostasis. And that's going to maintain a stable internal environment within our body. Everything works so that everything works. Now, metabolic reactions of the body, they're going to produce byproducts. And some of these byproducts are useful and recycled. Now, other byproducts are not useful and can harm these and then are going to be excreted out of the body. So, some examples of the waste products. We have CO2 from carbohydrate and fat metabolism. We also have nitrogenous waste like urea from protein metabolism. Note that urea is a waste product of protein breakdown from the liver specifically. It's carried through the body, it's filtered through the kidney, and ultimately it's eliminated in the urine. Now, bile salts and bile pigments from red blood cell breakdown is another form of a waste product. And then we have other various salts and tissue breakdown that's occurring. So, what other systems help waste get out of our body as well? Well, remember our lungs, through respiration, are going to eliminate CO2. Our integumentary system, also known as our sweat glands or skin system, that's going to help eliminate water, electrolytes, and salts. Our digestive system is going to be like our intestines, and that's going to help eliminate bowel pigments, heavy metal salts, and food waste. Like food waste in the colon. And then our urinary system. Through urination, we can eliminate urea, salts, and water. So, parts of the urinary system. We've got 2 kidneys, 2 ureters, a urinary bladder, and a urethra. Again, that function, maintain homeostasis. We're going to filter things, we're going to reabsorb, then we're going to secrete some things. We're going to keep the acid-base pH of the body homeostatic and help keep it balanced. And then we're also going to produce some hormones, along with regulating our blood pressure. So where are the kidneys? Well, they're located in the dorsal part of the abdomen, just ventral to and on either side of the first few lumbar vertebrae. They are also retroperitoneal to the abdominal cavity. So they're between the peritoneum and the dorsal abdominal muscles. In most domestic animals, the right kidney is more cranial than the left. So surrounding the kidney is a thick layer of peritoneal fat. That's going to help, just like we have fat around the heart, it's going to help protect them from pressure excreted by surrounding organs. Okay, looking at these x-rays, we can see, let me get my clicker. We can see that the spine is running this way, pelvis is down through here, there's the femur. We then can't see our diaphragm, but this is our stomach. Okay, this is over here. But what's cool is we can see a kidney and then another kidney. We can also see a urinary bladder within here. Now for this specifically, why this is colored, why these are kind of a little more colored too, and why this is flowing through here, it looks like in this picture there must be some sort of urinary contrast. A contrast is just like a liquid substance that's been given to this animal to highlight visually on the x-ray where things are. Over here at this picture, we can actually see a kidney here and a kidney over here. Now, let's talk about the gross anatomy or the structural anatomy, what we can see. We can see that it's a bean-shaped in most animals. It has like a reddish-brown color. It's covered by fibrous connective tissue capsule. And then, looking a little bit further, we can actually see the hilus. It's indented area on the medial side of the kidney. Now, it's the area where blood, lymph vessels, nerves, and ureters enter and leave the kidney. This hilus is a very important door that all these things go in and come out of. Now, then we have the renal pelvis. So if we cut a kidney in half, we could see the renal pelvis. It would be funnel-shaped area inside of the actual hilus. Now, urine collection chamber is going to be present there, and that's what that renal pelvis helps with. And that's going to form at the beginning of the ureter. It's lined with transitional epithelial cells, and that's very important to know because it's going to be stretchy tissue. And what else has stretchy tissue in the urinary system? Well, that's actually going to be your bladder. And sometimes we can actually see transitional cells in urine samples from the renal pelvis. But we'll cover more of that in Lab Techniques 1. Just know that it's stretchy. Oh, but why is it important that it's stretchy? Well, has your bladder ever been like really, really full or really, really small? Yeah, that's because of those transitional cells. It's moving, it's stretching for you. And then it talks to your brain and says, hey, gotta go. All right. We also see a renal cortex, which is going to be the outer portion of the kidney. We then can see a renal medulla, which is the inner portion around the renal pelvis. We then see calyces, or a calyx. This is going to be cup-like extensions of the renal pelvis into which the we have medullary pyramids, and they're going to fit kind of in there. So if you can see from this one visual, let me grab my pen, we can see the calyces are actually going to be within here, okay, kind of in those areas, and it's within the medullary pyramids, which are then also these as well. Now, the calyces act as funnels to direct fluid into the renal pelvis. What's also really interesting is that the kidneys have different formations depending on what type of species you are. So, for example, a horse. Well, food for thought, the right kidney is compressed and can appear heart-shaped. They must love their kidney. Also, in a horse, they have medullary pyramids, but they're going to be unipyramidal, okay? Meaning only one pyramid. But then we look at a cattle and we see that there's actually no distinct renal pelvis. So a cattle is going to be this B section. So we have a renal pelvis. If you look at top A, we have a renal pelvis here, but we do not have one down here for a cow. There's also going to be 12 lobes. So boing, 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 12. And it's going to be lumpy bumpy, like we can see here. And it's going to be multi-pyramidal, okay? So here's a pyramid, here's a pyramid, here's a pyramid, 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 okay? But for a horse, it just has one pyramid, kind of like down here in C. But that's not a horse one. Pigs are also going to have multi-pyramidal kidneys as well. You have a multi-pyramidal kidney, congratulations. But guess what doesn't? A canine and a feline. They are going to be unipyramidal, like we see in picture C. Okay, how do you know when you need to go? Feel free to go ahead and watch this video. It talks about how the bladder, the cells, the parasympathetic nervous system, it's all correlated to the pons in your brain that tell you when you need to go to the bathroom. Now, where does urine come from? We're going to cover that. Think microscopically. Let's get small. Okay, the microscopic anatomy. So why are the kidneys so amazing? Well, they're amazing because they have this cool thing called nephron, okay? And it's the basic functional unit of the kidneys. Now, it's not like they have one nephron. No, no, no. They have hundreds to thousands of microscopic, you have millions of nephrons. A cat has about, per kidney, 200,000. Dog has 700,000. And a sheep, it has like 1,000,000 per kidney. It's pretty impressive. Now, what does a nephron do? Well, it's going to filter out blood, it's going to reabsorb molecules that we need, and it's going to secrete what we don't need. All right, so looking at this picture, take time to just kind of see all of the different structures that we talked about, your cortex, your medulla, your calyx, your renal pelvis, your hilus or your hilum. We also have this renal artery. So artery is away from the heart. So this is taking oxygenated blood and going into the kidney. And then a renal vein that's going to, after our arterial oxygenated blood goes through our kidney, it's going to do some fancy things that we're going to talk about, and then it's going to deoxygenate and take that deoxygenated blood to the renal vein and to the caudal vena cava. We then have a ureter. Let's see. All right, we're going to stop there because then we're going to move on to what's inside those pyramids, what's inside the cortex, medulla, and the glomerulus. Okay, so what's in a nephron? Well, it has a renal corpuscle. It has a proximal convoluted tubule, or you may see this abbreviated as PCT. It then has a loop of Henle. It's going to have a descending or downward loop of Henle and an ascending going upward side. And then it also has a distal convoluted tubule and then collecting ducts. Now, different nephron structures are going to be in different locations of the kidney, okay? So, the nephron is going to pass through the cortex and the medulla, okay? And that's very important to remember because we're going to have absorption and secretion take place in different aspects of the nephron, which are in different aspects of the cortex and the medulla. And it's very important to know because certain functions of that nephron cannot take place without being in that certain area. Okay, so the renal corpuscle is located in the renal cortex. It's made up of the glomerulus, the Bowman's capsule, and then within that Bowman's capsule, we then have the Bowman's space, which is the space between the Bowman's capsule and the glomerulus. And its function is to filter blood in the first stage. So like as blood enters the kidney, goes into the nephron, this starts the first stage of how we're going to produce urine. Now, the renal corpuscle houses the glomerulus. Like we said, it's made up of those three different things. The glomerulus, and this is where you should get out your visual aids, is basically like a big old ball of capillaries or a tuft, okay? It is surrounded by the Bowman's capsule. The Bowman's space, we then see, is the space between the tuft and the ball, or the glomerulus, and the Bowman's capsule. The purpose of the renal corpuscle is to filter blood, and then that fluid portion of the blood is what's left after it's done this process is called the glomerular filtrate. Okay, so let's think about the blood as it comes in through that renal artery and the flow. The blood's going to come into the kidney, it's going to get into the hilus, it's then going to get into the renal artery. From there, it's going to go to the afferent arteriole, and that's going to take blood to the glomerular capillaries or the renal corpuscle. And then we're going to go into the glomerulus, where it's going to filter the blood, then going into the Bowman's capsule, and then the Bowman's space. So some molecules are going to pass into the space, and some are actually going to come out the efferent arteriole, which we'll talk about that later. So we look at what's going to get pulled out of the glomerulus is now going to be called the glomerular filtrate, okay? And now that then travels through the PCT. Here is a microscopic view of a glomerulus. That would look kind of cool. So here's all your glomerulus, all your tufts or your fluffs, big ball of joy. And then the afferent arteriole comes through here, and then the efferent is going to be coming out through here. So it's just a cool view. In class, we took a 10-minute break. All right, moving on. We have the proximal convoluted tubule. It's located within the renal corpuscle. It's a continuation of the capsular space of the Bowman's capsule. It's going to be the space that runs into the proximal convoluted tubule. So we've got the Bowman's space, and then it runs into the PCT. Now it's proximal because it's closest to the renal corpus, and then it's convoluted because it's complex and twisty and windy, as we can see from these pictures. It's also going to be lined with cuboidal epithelial cells, and that's going to have a brush border. Where have we heard brush border or microvilli before? It's going to be the digestive tract. Okay, so these are going to have microvilli as well. And the reason they have that is so that they can have the absorption, distribution of nutrients and all that. Okay. We then can see within this twisting path through the cortex. Okay, so the twisting part and the moving around and all that PCT, that's going to be in the cortex. And that's going to have a lot of reabsorption occurring. The glomerular filtrate, that's now called, once it's going through there, so we go to glomerulus, Bowman's space, glomerular filtrate. Now we're going to be, because we're in the tube, we're going to be called the tubular filtrate. And the brush border is where the reabsorption is going to take place. So the microvilli on the cuboidal epithelial cells within the PCT are going to sway back and forth and help with reabsorption. Reabsorption of things like calcium, potassium, and so much more that we'll talk about. Now let's move the tubular filtrate through the PCT, and we're going to reach the loop of Henle. Within the loop of Henle, there is a descending. In this picture, we see a descending and an ascending limb. It's going to descend from the PCT into the medulla. Okay, so look right here. There's the cortex and the medulla. So the PCT, Bowman's capsule, glomerulus, all this is up here. And then once we cross over into the medulla, starts our loop of Henle for the descending. It's then going to turn down here, hairpin turn down here, and go up through the ascending loop of Henle. And that's going to then go back from the medulla into the cortex. The descending loop has cuboidal epithelial cells similar to the PCT. Now, cells are going to be present at the bottom of the loop down here, okay? And those are actually going to be epithelial cells. They're going to be flattened to simple squamous epithelial cells, and they lose their brush border. Okay, so think about it. Within the descending loop, okay, there is the cuboidal cells. Once you get down to the turn, or the loop down there, basically, those cells are now going to be simple squamous epithelial cells. They're not going to have a brush border. And then, once we go back up through the ascending, those cells become thicker again, and they're going back to the cuboidal epithelial cells. Okay, so think about it on a racetrack. You drive really, 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 really fast on a racetrack when you're going straight, and then when you're taking a turn, you're going to kind of go at a slant or an angle, and that's going to thin out your turn and your strides a little bit. So you've got on the sides when you're running straight, you've got cuboidal cells. And then when you're making those turns, it's going to flatten out that stance and flatten out those cells and make that change. I don't know if that helps. All right, moving to the distal convoluted tubule. This is now going to take our tubular filtrate, and it's going to continue to travel through the loop of Henle, but now it's going to go into the DCT, which all that is in a continuation of ascending loop of Henle. It's not as convoluted, so it's not as twisty-windy, moving around. And it's distal to the renal corpus, so that's why they call it distal. And remember, it's located in the renal cortex. We then can take the DCT, and it's going to be from all the nephrons in the kidney, are going to then empty into the collecting duct. So all these nephrons, all the nephrons go into the collecting ducts, okay, as we can see from this picture. Now it's going to carry the tubular filtrate through the medulla into the calyces, and then it's going to empty into that renal pelvis. So we go out and we go back in. We go out, and through the collecting duct we go back in. It's going to regulate our urine volume. It's going to be also the primary site of the antidiuretic hormone. Remember that endocrine system? And the regulation of the acid-base balance. That's where this is going to happen. And once it's in the collecting duct, that tubular filtrate now becomes urine. All right, we talked about the renal artery and then how it goes, the blood will go to the afferent arterioles, right? And then we go to the glomerulus and so on and so forth, all the things that we just said, okay? But remember how I said it's like not all of that blood is going to be filtered and used through there, okay? Because we also have to oxygenate our cells and structures of our kidney and our nephrons, right? So where does it go? Well, then it goes to, instead of the afferent, so basically your blood's going to come through renal artery, goes through the afferent arteriole. Your body's like, all right, I'm going to take what I need right now from the blood. And then what it doesn't need, it then can go back through the efferent arterioles, where it's going to divide to form the peritubular capillaries. And that peritubular capillaries, or that structure, is going to surround the nephron. And it's going to take oxygen and transfer it to the cells of the nephron. Okay, so that takes place. the tubular reabsorption and secretion is also going to happen here. So these peritubular capillaries, they coverage to form the venules, then they make larger veins, and finally, they're going to be in that renal vein. Okay, so arteries away, vein is going back to the body and is going to enter the caudal vena cava. Here is a video to show the renal blood flow, but we're going to move on. All right, in class, we kind of went ahead and did a breakout, and we went through and talked about the functions of the kidney. You guys can look through this and do this on your own if you'd like. We broke apart the parts of the nephron. We talked about reabsorption and secretion. We did reviews of the urine formation and all that, as well as the renal blood flow review. So if you're able to kind of do this on your own, then you did great. All right, this also is an example of the urine formation video. Okay, now, stuff that we did not get to cover in class that is very important. As you know, we talked about the reabsorption that takes place with our kidneys. So now let's break that down just a little bit more. You've got your notes. There's also some visuals to help with this. This can get a little sticky, so feel free to pause and go as you need. So we talked about how blood comes through. It can either go to the afferent or efferent. So it's got kind of two options here. Now, substances can go into the Bowman space or the peritubular capillary bloodstream. Now, substances are going to be reabsorbed. They're going to pass out of the tubular lumen through or between the tubular epithelial cells, like we talked about. Now, the substances that are going to be reabsorbed are then going to enter the interstitial fluid, and they're going to pass through the endothelium into the peritubular capillaries. Okay, we just talked about this. So reabsorption is equal to substances getting back into the bloodstream. Did you know that 65% of all reabsorption takes place in the proximal convoluted tubule? And that 80% of sodium, water, chloride, bicarb are going to be reabsorbed. That's a high percentage. Did you know that 100% of glucose is reabsorbed? And that's very important because where else would it go? Our urine. And is glucose supposed to be in our urine? Absolutely not. Also, the loop of Henle, the distal convoluted tubule, the collecting ducts are also going to facilitate the reabsorption. And the loop of Henle and collecting ducts, this is where urine is concentrated. So water recovered from urine to reabsorb back into the blood. That's where that's going to take place. Let's dissect more. Okay, so you've got these fun guys like potassium and calcium. That reabsorption is going to take place in the proximal convoluted tubule. Like we said, 80% of those are going to be there. Now, we're also going to see it in the ascending loop of Henle and the distal convoluted tubule. So calcium is going to move from the filtrate under the influence of vitamin D and, remembering our endocrine system, our parathyroid hormone and calcitonin. Now, magnesium is reabsorbed from the proximal convoluted tubule through the ascending loop of Henle and the collecting duct. And that parathyroid hormone release is going to increase the reabsorption of that magnesium. See how everything is connected. Everything works so that everything works. Hashtag homeostasis. Okay, now sodium. Sodium gets its own slide. So sodium is a tubular filtrate that attaches to carrier proteins that move it into the cytoplasm of the proximal convoluted tubule epithelial cells. Okay, remember there's cells within the PCT, and those cells have nucleuses and cytoplasm and all that. So glucose and amino acids, remember proteins break down into amino acids, sugars break down into glucose. So glucose and amino acids are going to attach to the same carrier protein. Okay, they're going to buddy up and follow sodium into the cell by passive transport. Okay, we've You should remember some of this through the sodium ion pump channels. So glucose and amino acid are like, hey, sodium, what are you doing? Can we transport with you? Remember that sodium is actively pumped out of the cell into the interstitial fluid, where it moves into the peritubular capillaries. So again, glucose and amino acids move from the PCT cell into the interstitial fluid because they're like, hey, sodium, can we hang out with you? And then into the peritubular capillaries by passive diffusion. So we have passive transport, and now we have passive diffusion. So sodium ions are reabsorbed in the ascending loop of Henle and the distal convoluted tubule. They usually are exchanged for like hydrogen and ammonium or potassium ions. Now let's talk about chloride. Chloride is going to diffuse from the tubular filtrate into the epithelial cells and the interstitial space in response to an electrical imbalance created by sodium removal. So some of the water in the filtrate moves into the interstitial space and the peritubular capillaries by osmosis. So once sodium, glucose, amino acids, and chloride have left the tubular filtrate is when this occurs. So we then have this thing called urea, which you should be familiar with. So this is one of the kidney's values, okay? We measure on blood work panels, okay? So we can check to see how much urea, which is also known as the blood urea nitrogen or the BUN, we can measure that value in a patient's serum or like a fluid portion of the blood. So the urea is the end product of protein breakdown, and it is filtered at the glomerulus into the proximal convoluted tubule. Most of it is going to stay in the urine, but a little is going to be reabsorbed through osmosis. Now, when I say it stays in the urine, it's not going to be reabsorbed through osmosis. It's because it's like with that urea buildup, it can cause toxicity. So we want most of it to stay in the urine so we can urinate it out because it's a byproduct of protein breakdown. So if you drink a protein shake, the protein molecules would get into your body through the digestive tract, it would break it down, and then what it's going to break down into is amino acid, and then the amino group separates and it makes a nitrogen compound or molecule, which then that can break down into ammonia and so on and so forth. So we don't want ammonia in our system. Okay, so we want the urea to still be excreted in some form or fashion in large amounts, so that way it doesn't build up in our system and cause toxicity. Now we have this thing called creatinine, or we can shorthand it with CREA. But creatinine is the breakdown product of muscle. It stays relatively constant in most animals. It is filtered into urine at the glomerulus, and none of it is reabsorbed. So it all gets urinated out. Because of this, it's a good indicator of glomerular filtration rate. So if our creatinine is going up in an animal, that means that the GFR is going down. So when I say the creatinine is going up in an animal, this is another blood work panel that we can run on our kidneys, and we can say, okay, what is our creatinine level at? Okay, that's one of our kidney values. What is our creatinine level at? And if it's elevated, then this means that the glomerular filtration rate, okay, that's the glomerulus working in that 1 minute, is going down. This means there's fewer functioning nephrons. It is an indicator of the rate and the degree of kidney failure. Okay, so creatinine is a great diagnostic test through blood. We can draw blood on an animal, run a chemistry panel. One of those chemistry values can be creatinine, which tells us kidney function, and specifically, it's how the glomerular filtration rate is doing. And if it's doing bad, then that might be a sign that our patient is in kidney failure. Now, when it comes to secretion, it's primarily going to occur at the distal convoluted tubule. Hydrogen, potassium, and ammonium are going to be eliminated by secretion here. Now, some medications are also eliminated from the body by the secretion in the DCT. This is a good visual to kind of look at the differences between the reabsorption and secretion taking place. Big note here, remember we are looking at the cortex and the medulla, and we have our PCT here. We then are going to come down through here, and then we have our loop of Henle. Remember those different sizes and different shapes of those cells and how they take place. And then you have your DCT and then out your collecting duct. Now note all the different, you don't have to memorize this, but this is just showing you that how and where those different minerals, such as your sodium chloride, your potassium, this HCO3 here, this is bicarb, your glucose. Remember, glucose and amino acids are like, hey, sodium, can we hang out with you? Okay. Down in the loop of Henle, you've got magnesium, you've got calcium. All these different molecules are going through either osmosis or passive or active transfusion to help keep the balance of your nephrons and your kidney and your body. All right, the volume regulation. So what regulates it? Well, it's determined by the amount of water contained in the tubular filtrate when it reaches that renal pelvis. Okay, it goes in, it comes out. It goes in, it does some cool things, it comes out. And it's controlled by the actions of the antidiuretic hormone from the posterior pituitary gland and also aldosterone. Okay, make sure you remember your endocrine hormones because yes, they're important. Now your ADH or your antidiuretic hormone acts on the distal convoluted tubule and the collecting ducts, and it's going to promote water reabsorption. Now, aldosterone is going to increase the reabsorption of sodium at the DCT and the collecting ducts. This is where we get the osmotic imbalance that encourages water to follow sodium out of the tubular filtrate and into the blood. Aldosterone is a key in potassium homeostasis. Okay, so your hormones play a huge role here. Okay, blood pressure. We talked about how does the kidneys and the nephrons, how does that relate with blood pressure? Well, we have this thing called a renin, angiotensin-aldosterone system. And that's when the afferent arterial cells release renin in response to the decreased blood pressure or our sodium chloride concentration. And renin is an enzyme which acts on the angiotensin. Now, angiotensin 2 causes arterial constriction and stimulates the aldosterone release, which is then going to cause an increase in sodium and water reabsorption back into the bloodstream, which is going to increase the blood volume. So in short, it increases in blood volume, it fills the vasculature better, and it increases the blood pressure. Okay, I do need you to know that there's the enzyme called renin that with hormone stimulation, okay, it plays a role with sodium and water reabsorption, and it wants to balance things out, and it can increase our blood pressure. And what's cool is actually within the glomerulus, the glomeruli, because it's, those tiny little afferent arterioles go into the glomerulus and all that, that actually pumps like boom, 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 boom. And it can speed up depending on the actual blood pressure within like the body and in the nephron. So that's pretty cool. Moving on to our ureters, they're going to leave the kidney at the hilus. Hi-bye. It's an outer fibrous layer. It has middle smooth muscle layer, and inner layer that's lined with transitional epithelium. Now that transitional epithelium is going to allow the ureters to stretch. Remember, that's a stretchy type of material as urine passes through them on its way to the urinary bladder. That's where we can see transitional cells taking place as well. It's going to have smooth muscle layers. It's going to propel urine through the ureter through peristaltic contractions. So there's peristalsis again. The ureters are going to enter the urinary bladder at an oblique angle, and when the bladder is full, it's going to collapse the opening of the ureter, like we saw in the video, and it's going to prevent the urine from going back the opposite direction into the ureter. So it's kind of like an upside down triangle, if you think about it. Now the urinary bladder, we've talked about it, is lined with transitional epithelium that stretches as the bladder becomes filled with urine. The wall of the urinary bladder contains smooth muscle bundles, almost like a blanket. The neck of the bladder is going to extend caudally from the sac into the pelvic canal, and it joins in the urethra. And around the neck of the urinary bladder are circular muscles composed of skeletal muscle fibers, just like we saw in that video. Now, we talked about in the video too, but here's just a refresher, the control of urination. So mictration, or uresis, is the expulsion of urine from the urinary bladder into the urethra. Urine accumulates until pressure of the filling bladder activates stretch receptors in the bladder wall. And then again, that spinal reflex returns a motor impulse to the bladder muscles, causing them to contract. Contraction gives the sensation of having to go to the bathroom or urinate. That's where then your voluntary control of the muscle sphincter around the neck of the bladder is going to result in temporary control of urination. So you can stop and start. Now your urethra is a continuation of the neck of that urinary bladder. It's going to carry urine from the bladder to the external, environment, hopefully the toilet or the woods. The proximal urethra is lined with transitional epithelium cells. God, they're everywhere, all those transitional epitheliums within this. And that's going to allow it to expand as urine leaves the neck of the urinary bladder. Now, the female urethra is shorter and straighter than the long curved male urethra. That is so important to know. Star that. So in the female, the urethra opens on the ventral portion of the vestibule of the vulva, or the vestibule of the vulva, the opening, the first compartment. And in the male, the urethra runs down the center of the penis, and it also functions in the reproductive system. Noting that the female urethra shorter, straighter than the long curved, that curvature of the male urethra, that anatomically can cause a lot of issues when we think about it in relation to crystals and mineralization, and is one of the components of why male cats are so prone to getting urinary blockages. Here are some additional resources. The crash course, you guys have seen those before potentially, or we've recommended them before. They really are such a great, fun, and visual way to reintroduce and review this stuff again. So hope you enjoy it, and let me know if you have any questions.